Welcome to some additional biology review. Hope you enjoy.
Let's take a look at enzymes now. First, let's look at the reaction. Now, the reaction will take place once the enzyme and the substrate combine at the active site. Once they combine, they form this complex. And this complex is known as the enzyme substrate complex. And this is actually where the reaction takes place. Now, once the reaction takes place, these products are made and released. And once the products are released, the enzyme is once again free to combine to another substrate. Sometimes there's these things that we call inhibitors, and they attach to the enzyme in the active site. In other words, they block the substrate from attaching to the active site. And because they won't be able to attach to the active site, they will not form the complex. In other words, the reaction will not be able to proceed, and therefore they won't be able to produce those products. Now you need to remember that the goal of an enzyme is to lower activation energy required for that reaction to take place. It's still going to need energy, but it's going to be much less. Therefore, the reaction will be able to take place faster. Okay, so if we look at these two lines in this graph, and they're illustrating how the reaction is progressing, you'll notice that this line right here is actually requiring less energy, and that's because it is using an enzyme. So activation energy with an enzyme. Now notice how much it would have been if you actually didn't have an enzyme. So this would be without an enzyme. Okay, so now let's complete the Venn diagram for passive and active transport. In the middle, we're going to discuss what they both have in common. Now, here is our word bank in the bottom. So let's start with the first one, which says ATP is required. Now we know that active transport is the one that requires energy. So we're going to include that one on this side. ATP required. Next, and we can cross that out. Next is down concentration gradient. Now, down can also be referred to as with the concentration gradient. So basically you're saying that you're going with the flow. You're not going to go against it, meaning that you would not require any energy for the resistance. So this would be passive transport, no energy required. So that's down concentration gradient. As for the opposite, which would be against concentration gradient, that would be active transport. You're going against the flow, you're going to have resistance, so you're going to need energy for it. So this would be against concentration. Let's cross that out. Next is facilitated diffusion. Now remember, diffusion itself is a form of passive transport. Now facilitated is referring to using a protein to help you be able to get the things across. So it's just making things easier with that protein, but there's no energy being needed. So that's going to be under passive transport. So we're going to write in facilitated diffusion. Okay, next is osmosis. Osmosis again is also passive transport, and this is the movement of water. Then we have endocytosis and exocytosis. Both of these are active transport. Endocytosis, exocytosis. Remember that ending cytosis. We're talking about active transport there. Simple diffusion would be passive transport because of that diffusion part. So simple. And this means that there is no proteins being used here, okay? But again, there is no energy needed as well. Then it says move material into and out of the cell. Well, keep in mind that they are both transports, and they are both transporting things into and out of the cell. So that would go under both. Move material into and out of cell. So let's scratch that out. Okay, a transport protein, protein channels, protein pumps. So now we're talking about the different types of proteins. Okay, so we could include a protein pump. We know that would definitely be under active transport. So that's protein pump. Now, the protein channels, they're simply an opening. It's simply a tunnel, okay? There's no magic little key to turn them on and off, which would require energy. So that's why protein channels, those are actually the ones that are used for facilitated diffusion. So those we're going to include them under passive. Protein channels. Now notice how both of them were using proteins to transport, which is why we would say any transport proteins can actually be under the both category. So this would be transport proteins. It just depends which type. Okay, and then we're left with high to low and low to high. So high to low means that that is the actual way to go with the concentration. So that's passive transport. So that would be high to low with the flow. Okay, low to high, that's actually going against the flow. That's something that is going to require energy. It's going to go through resistance. So that would be low to high. All right, and that's it. Okay, so next let's take a look at protein synthesis. So this is the process known as protein synthesis. And protein synthesis has two steps, step 1 and step 2. Step 1 is where you're going from DNA to mRNA. We call that step transcription. Step 2 is going from mRNA and determining what the actual amino acids would be based on those codons, and that would be known as translation, because you're translating the code. Okay, now this polypeptide, remember, is made up of many amino acids. All right, so let's look at this example. Now, remember, DNA and mRNA are complementary. The only difference is that you do not have any T's in mRNA. Instead, you have U's. So let's look at what the complements are. T pairs up with A pairs up with U, C with G, T with A with U, G with C, A with U, G with C, C with G, A with U, T with A, T with A. Now looking at a codon chart, we would know that AUG codes for met, AUC codes for isoleucine, UCG codes for leucine, and then UAA is a stop codon. So let's talk about viruses. There are two cycles that you need to be familiar with for viruses. There's the lytic and the lysogenic. Now, the lytic, you can think of it as a shorter cycle. Lysogenic takes much longer. At the same time, we often refer to the lytic cycle as the active stage and the lysogenic as the dormant stage. Now, with these viruses, the first step that's going to be seen is where the virus attaches itself to the host cell and injects its DNA. Now, once the virus has injected its viral DNA into the host cell, this is the host cell here. now it has the choice of going through the lytic cycle or the lysogenic. Now, if we're focusing on the lytic cycle first, we have the viral DNA and the host cell's DNA. The viral DNA is actually used through protein synthesis in this case to create or to produce viral components, and these components are basically viral proteins. Now you need to understand that these proteins are being produced with the host cell's machinery. So with host cell machinery. In other words, a virus cannot produce these proteins on its own. It requires that host cell. Now, all of these proteins that are being developed are actually the components that are going to be used to make more viruses. Then, once it has enough of those components and starts to assemble those viruses, we call those new viruses as virons. Okay, and once too many of them are building up here, then that's when the cell bursts, also known as lysis. Okay, so that's when the host cells lyse. Now, that is the lytic cycle. And as you can see, once these virons are released, they have the opportunity now to go and attach themselves to another host cell and repeat the process once again. Now, if we are focusing on the lysogenic side on the other side, this same thing happens. The virus will attach itself to the host cell. So this is step one. Then that's when you end up having the viral DNA and the host DNA together in the same cell. But then from here, instead of it right away starting to produce those proteins, those viral proteins, instead, something really strange happens here, which is seen in step 3 here, where the viral DNA actually integrates with the host DNA and it becomes part of it. Now, and as you can see here, the red component, which was symbolizing the viral DNA, is now part of the host DNA. Now, what's so special about this is that when you go on to step 4, which you see here, the cells will continue going about their regular cell cycle. So here we see the cell cycle, which is what's going to lead to cell division. And if you remember from our talk about cell cycle and cell division, in order for the cells to divide, to make more cells, they need to replicate DNA. Well, that DNA is no longer just the host cell's DNA. It also has that viral DNA in it. So that means that it's going to replicate that viral DNA as well and have more cells that have that viral DNA. Well, eventually, it just continues to do that over and over again. And eventually, for whatever reason, something triggers that virus to become active again, and it no longer is dormant, okay? And it becomes active. And when it becomes active, it can actually go into the lytic cycle and produce those proteins, which will eventually develop those virons that will burst that cell or lyse the cell. So make sure that you understand the difference between the two cycles and understand which one is known as the active and which one is known as the dormant. Are different levels of taxonomic classifications. The first that we're going to mention is the broadest, and then we're going to move down to the most specific. Now, there's a great mnemonic way to remember what these levels are called. So we remember it as, did King Philip come over for great soup? Now, if you remember that mnemonic device, then it will be easier for you to remember what these levels are called. Now, the first one for did, that is the domain. And remember, that is the broadest. Then we move down to kingdom, phylum, class, order, family, genus, and the last, which is the most specific, would be species. Understanding the hierarchy of taxonomic classifications will be very helpful when you're looking at the binomial nomenclature, which is basically a two-word naming system. Now, this system consists of a capital and a lowercase letter. Sometimes these are underlined or they are italicized. Overall, you need to understand that the capital letter represents the genus and the lowercase letter represents the species. Look at the levels of organization, starting from the smallest level, which is the atom. The atom makes up molecules. Molecules make up cells. Cells make up tissues. Tissues make up organs. Organs make up organ systems. Organ systems make up organisms. A group of the same type of organisms make up a population. Several populations in the same area make up a community. And several communities within the same area or region make up an ecosystem. And various ecosystems make up the biosphere, which is the largest levels of organization. Before we begin by trying to figure out what those 10 missing arrows are, let's first review what the arrows symbolize. Now, arrows point towards the direction of energy flow. In other words, they are pointing at how is it that the energy is flowing from one organism to another. So in this case, we see that the seeds would be the producers, and the energy from the seeds is flowing in the direction towards the grasshoppers when the grasshoppers are eating it. Okay, then we have the grasshopper that is getting eaten by the frog, which is then getting eaten by the hawk. Now, as you see, you can follow those arrows in that direction. Now, there are 10 missing arrows, so let's try to figure out which ones are missing. Now, we have another producer over here, which are the berries. And the way that I can tell that that's the producer is that all of the arrows are pointing away from it, meaning that it is getting consumed by other organisms. For instance, in this case, the berries are getting consumed by birds, by grasshoppers, by aphids, but notice how it could also get consumed by a butterfly, which then would get consumed by a frog and then a hawk and so forth. So that would be one of our missing arrows. So let's change the color here and we're going to make that red for the missing arrow. So that's one. We still need 9 more to go. Now, if we continue this flow, we see that berries can also get eaten by aphids, which get eaten by ladybugs, and then ladybugs can get eaten by birds, but they can also get eaten by dragonflies. Then the dragonfly can get eaten by a frog. So there we have 3 missing arrows. Let's keep going. Now, the frog can get eaten by a hawk, but it could also get eaten by a snake. Now we know that snakes can get eaten by hawks. Okay, so then we have found 5 of them so far. Let's keep going. We need 5 more to go. Now, we know that the snake can get eaten by a hawk. The hawk can also eat birds, but the hawk can also eat mice. So this mouse can get eaten by the hawk. The mouse can also get eaten by the fox. The bird can also get eaten by the fox. And the rabbit can also get eaten by the fox. Now, what else do rabbits eat? Well, rabbits, we know, consume producers. Okay, so take a look at the parts of a flower. Now, first we're going to be looking at the female reproductive parts. And the female reproductive part, the entire structure itself, is called the pistil. And it consists of the stigma, style, and ovary, which consists of the ovum. Now, the male reproductive structure consists of what we call the stamen, and it has at the top the anther, and this is the one that holds pollen. And then what's holding the anther we call the filament. Now, in the back, what gives the flower those really beautiful colors, for instance, these right here in the back, that is what we refer to as petals.
This concludes our review. Make sure to study and have a great one.